What's up, everyone? Today we're talking about molecular motors, and these are intricate, really huge proteins that are responsible for some really cool stuff that we'll talk about. Now, by now, you should understand that proteins are really one of the building blocks of life. And we talked about enzymes, and we love enzymes. Enzymes are intricate, they are very sophisticated, they are responsible for most of the metabolism, and they are very well regulated. And they do some really cool stuff, right? So let me look at an example. Here we have kinase, and kindness is pretty good. It has an intricate structure, and we know that they do phosphorylation, and you have a lot of these. So this is a good enzyme that we like, but I want to show you something else. So look at this thing right here. Every one of these little one components like looks like a kindness. It's as big as a kindness. Like every one of these is as big as a kindness, but look at how intricate it is. Look at all of these. And these are proteins. These are, helices. And look at how intricate it is. This is crazy, right? And this is just the beginning. And I'll tell you what this thing is by the end of this video, but you can go ahead and guess and see if you can guess what it is. But we'll talk more about it later. Now, we are talking about things that we call molecular motors. All right. So the molecular motors, right, are these proteins, or just, it's not really proteins. They are proteins, but they are not enzymes, and they are not just peptides, right? These are just really intricate components. They are intricate complexes, right? You saw how that one has a bunch of complexes that work together to do something that looks like a motor, right? Think of a motor for your car, or a motor that turns things. All right, so these are examples that we're going to look at today. So one is the molecular mechanism of muscle contraction, right? You might have heard of actin and myosin and how they work together. That's a molecular motor. And another one is cytoskeleton, right? The things that keep the cell intact, pretty much. So we have microtubules, tubulin, kinesin, and things like that. You have molecular motors that unwind DNA and bacterial flagella and stuff like that. Now, these molecular motors come in two types, right? Two types. What are the two types? One is the linear motors, and the other one is rotating motors. So it's two types. So the actin, the myosin, these are linear. The tubulins are linear. But when you're talking about the DNA helices, or GTPase, ATPase, if you heard of it, in the electron transport chains, these are even better motors that rotate. So these are the two types of motors that we're going to look at today. All right. And I think I have this here. Again, two types. One is a rotor. The other one is, no, this is for rotating parts. But again, we have linear motors, and then you have rotating motors, right? And rotating motors, of course, they have a rotor and a stator. A rotor is the one that turns, a stator is the one that doesn't turn. All right. And again, these motors, when you hear motors, what you hear? Motion, right? You hear motion. So these are used to move, whether it's rotating things, whether it's moving things from one place to another. These are motors that are used to turn chemical energy into motion in the cell. That's mainly how they work. right? And mainly they work with ATP, but some of them use other things other than ATP. So again, linear motors that we're going to look at, kinesins, dyneins, myosins, helicases. The one that we're going to look at in this video is helicase and kinesins. Rotating motors that we're going to look at is we're going to take a little bit a close look at the flagellar motor and not the ATP synthesis because we're going to have a whole chapter on this. So we're going to look at DNA helicase instead. But again, this is just an introduction. This is just to show you how cool they look. Maybe tell you how they work a little bit, but we're not going to talk too much into the mechanisms and stuff like that. So let's start with muscle construction. So have you ever thought about the things that happen for you to contract your muscle? Right? For you to contract your muscle and have this force and this energy, where does it come from? What happens? And what happens is, of course, proteins. What proteins? Myosin and actins, right? These ones are proteins, but they look at as filaments, really, right? Filaments of myosin and actin. How do they do it? They slide against each other, right? They slide against each other. So let's look at the graph that shows it better. So this graph, if you look down here, oops, make sure that you can see this. If you look down here, right, you see that you have one component that we call a myofibro, right? This is one component of the muscle. If you zoom in here, that's where you will find those filaments. What filaments? The myosin and the actin, right? And then look at what happens. Around the muscle, you have what we call sarcoplasmic reticulum. And why is this important? It carries calcium ions. And the calcium ions, when they get into the muscle, these are the ones that activate your muscles, right? So a nerve, so your nerve is going to carry a signal to activate a muscle, and then calcium ions are going to be dropped. So when you zoom in a little further, these filaments are going to look like this, and this is how the muscle works. So the filaments are going to become shorter and longer depending on the signal. That's how muscle contraction works. And let me see if I can show this a little better. Hopefully we can make this work. Yes, we can. Maybe. Yes. All right.
Muscle contraction is at the basis of all.
So if you take a muscle filament and you zoom in and you zoom in, you're going to look at the two filaments, actin and myosin. And how do they work together? They contract and then they become shorter and longer, just like that. And that's how muscle contraction happens, right? And then we're going to zoom in a little further and see what causes that contraction or that shortening and lengthening. And this is calcium, right? So the calcium is dropped in, and then on the actin filament, you have something called troponin that's going to be activated, right? Which is going to carry along tropomyosin, and this tropomyosin are going to, these are the ones that bind to the myosin. So top is actin, bottom is myosin. So these tropomyosin are going to make the binding sites available, and then we bind. The two bind. And when we bind together, that's pretty much where the reaction is happening, right? When the reaction happens, that's where the ADP is dropped. They carry along here, and this is when the ATP comes in. right? So this is how your ATP works for the muscles. The ATP in the cell is going to come in and bind so that it can release and bring it into the original state, right? So myosin ATPase is going to release ATP, turn into ADP. This is where the energy is used, right, to bring your muscle back to the original state, and that's the whole contractions. This reaction that I just showed you is going to happen again, and the muscle contracts and detracts back and forth, right? This is how it happens, and these two molecules work together to do this, right? That's really cool, and this is one of the ways these proteins work together, so that you can use your muscles. Now, let's go back to the cell. If you look at the cell here, this is a typical textbook cell. It has a certain shape. It looks really, really nice. And they show you all these organelles in the cell, but they don't tell you, at least they don't talk too much about why it has this nice little round shape. How is it shaped this way? The organelles are not responsible for it. It's the cytoskeleton, right? You have a lot of cytoskeletal filaments that keep it intact. So in fact, it's not empty like this, right? It's going to have a lot of networks of these filaments throughout all the organelles, but also shaping it all around the cell. So if you look at around the cells, you're going to have some that are all around it, like a scaffold around, but also again inside you're going to have them as well. What are those? Well, microtubules, right? And then actin filaments as well, and then some other intermediate filaments, right? But again, these are they make what we call the cytoskeleton. Outside, inside, you have the cytoskeleton, right? And again, you have different things that the cytoskeleton does, as you can imagine, right? So some of them, they establish the cell shape as expected. Some of them provide mechanical strength, just like the ones that you see in here. Some of them facilitate the cell movement, right? So you imagine a cell need to move from one place to another. They actually literally contract and they contract themselves, kind of like the muscles, but these are not really muscles. But cell motion is made by these fibers contracting and relaxing. Some of them support intracellular transport, which is really cool. We're going to talk about it in a minute. And some of them, you might have seen them in cell division. You've seen those meiosis and mitosis graphs that show you the filaments. So this is what the cytoskeleton does. What is a cytoskeleton when you look at it chemically? These are going to be networks of protein fibers, right? So these are going to be proteins, just like we've been looking at proteins, and they are going to have your amino acids and things like that, which is pretty unbelievable, but that's how it happens, right? So this is another example of these networks. But let's look at one in particular, which is the microtubules. Microtubules are polymers of a protein called tubulin, right? So it's tubulin heterodimer, meaning that you have two dimers of tubulin. I actually worked with tubulin. I did research with people at the University of North Texas, and I was working with them to target tubulin with different small molecules, but that's another story. But tubulin polymerizes, becomes microtubules, and these microtubules, they not only form networks, so these are networks of microtubules, but they not only make structural features, but they can provide like superhighways, which is really cool, right? So look at these microtubules here, they can form superhighways that are used to transport a micromolecule from one place to another. And if you zoom in, you're going to find things like this on top of the microtubules, and these are going to carry literally cargo across the cell. And what protein is responsible for that? Kinesin. Kinesin is another motor protein. And if you zoom in, it's going to literally be walking along. It's going to be walking along, and by that walking is ATP and the ADP reactions. ATP and ADP reactions are going to be, ATP comes in, moves it, ADP is released, ATP comes in again, and so on and so forth, which is really, really, really cool. Let me see if I can play this to show you how this works. And this video has other things, talk about motor proteins in general, which is pretty cool. So I just wanted to show you kinesin. So kinesin, the way it works again is going to carry cargo across microtubules, right? Kind of like a highway. So I just wanted to show you this nice figure that researchers has found. So yeah, this is the reaction. So this is how they found out that it looks like it's walking along the microtubules, but it's just a reaction, and this is kind of like how it looks like. All right, but again, this is a real microscopic thing, right? This is a real microscopic thing that shows the kinesin protein carrying cargo, right? And now this is in the lab, so it's carrying a plastic bead, but it's carrying it along the microtubules like a highway, like a car carrying things on a highway. It's really, really cool. So that's kinesin. Kinesin is really one of those coolest motor proteins that we like. So those are examples of these linear motor proteins. Let's look at an example of a rotating protein. So one is DNA helicases. DNA helicases is another one that does it. That's really cool. Again, we're gonna have chapters about this, so I'm not here to talk about how DNA helicases work. But one of the biggest parts in DNA replication is separating the two strands of DNA, and the way we do that is we use helicases, and the helicases are really, really cool because this is how they look like, right? It's an assembly. You see how it looks like? So it's an assembly here that of all these are hexamers, but sometimes they become dodecamers, which is 12 of these, and they form this nice circle. And you see this circle right here, this is where DNA comes in. DNA goes through this, and it's opened, right? So this is an example of how that happens. And let me see if I have a animation of this. I'll just play you an example of helicases. I won't talk about the whole thing. Yeah, there you go. So you see helicases here. You see it here, unwinding the DNA over there. So it's unwinding the DNA, and it has to unwind the DNA for everything else to happen. So you have DNA polymerases and all these other proteins that we'll talk about when we get to DNA replication. But the helicase part is really, really interesting. And this is another one of those really cool motor proteins, right? That is a rotating motor protein. All right. Another rotating motor protein that I don't have here that is very interesting is the ATPase in the mitochondrial membranes, right? The ones that make that is used to make ATP, right? You might have seen it, and it's also another rotating motor, right? And it's driven by what, if you remember correctly, it's driven by the gradient of H plus, right? So the H plus binding makes it turn the gear, right? It's literally like a motor, which is really cool. But another one that I wanted to talk to you about today is a bacterial flagella. It's another one, right? So the reason why I wanted to show you is because you've seen flagella in a lot of places here, and I wanted to show you that it's also a protein, right? And in this one, it's a proton gradient that drives this fragile motor in E. coli, right? So how many proteins? At least 25 proteins, and it's not just 25 proteins. It's 25 proteins, but some of them have dimers and trimers and tetramers. So it's a lot of them. So you have a rotary motor anchored in the inner bacterial membrane. You have a long filament that serves as a propeller because, of course, it's a flagella, and then you have a hook that connects as in universal joint that connects the motor with the filament. Right. This is a really cool flagella, and a few things. I won't talk too much about it, but I'll tell you that the ring, right? The ring again. You have a rotor and a stator, like we talked about, right? And the rotor portion, the ones that turn, it's made of several proteins, but the three known ones are these three: flig, phlegm, and flin. And then the stator portion is made by these two main proteins, MOT A and MOT B. But you have a lot of other proteins that are involved, so we won't talk too much about them. But let's see some of these in action. Come on, come on, come on. All right, so a lot of these, you see them everywhere. By the way, the human sperm also has a flagellum, but you might have seen these, but they are really really big, and they are giving you an example. They are giving you an example of how huge this thing is. So your normal, GPCR, which is a big protein, is really big, and then you have ATP synthase that we talked about. It's really big, but look at how much bigger this thing is. It's massive with all of these proteins. So these are the flav proteins that I was talking about, and flig, and then you have flin, and then phlegm. The other ones, these are the ones that are made that make your rotor, right? And then in the stator, you have these proteins as well. And in most of the time, again, this is chemically controlled. So here, this is a proton. This is a proton gradient, and the proton gradient is going to come. These are amino acids, and then the gradient is going to actually influence the rotation, right? And that's pretty much how it works. And this is one of the biggest proteins that we've seen, right? So again, going back to this one, I promise that I'll tell you what it is. Well, this is the bacterial motor that I showed you, right? This is the bacterial motor, and as you can see, it's just crazy. It's massive, and it's many many times the other proteins that we've been looking at. It's actually insane that somebody has been able to solve the structure of this, because I've worked in the lab. I worked with like one little filament of this, and to solve the structure of it took months. So it's crazy that somebody has solved the structure of this. And the way they solved it, by the way, is you have to solve one part at a time, because it's very hard to assemble this and purify it intact. But some people have done it, which is really good. But these are the proteins, and you can literally download this. You can analyze the amino acids. You can look at the patterns. You can look at all of this and change whatever you want. But this is the bacterial mother of E. coli, and that was mothers. Hopefully at the end of this, hopefully you have an even bigger appreciation of proteins, peptides, enzymes, and you not only know that they catalyze reactions and things like that, but you now know that these are the things that hold together our cells. These are the things that are involved in protein motion, but they're also involved in cell division, and many, many more other things. So hopefully that was pretty interesting, or at least as interesting as I find it. And I'll see you next time.